Good evening and welcome to Test 4, Cellular Respiration and Thermoregulation. The bulk of this test will consist of questions based on the cellular respiration section. We will do aerobic and anaerobic respiration, as well as some thermoregulation between endotherms and ectotherms. So let's start with cell respiration. Here, the goal is to produce ATP, and we'll find out later that aerobic respiration is going to generate somewhere in the 30 range of ATPs, 36 to 38, and anaerobic is going to yield 2 ATPs. But either way, what the cell will do with that ATP is hydrolyze it. Now, in case we forgot what hydrolyze means, that means to break, and to break with water. And what happens is if you take the ATP, which we know has 3 phosphates, you break it down to ADP plus the phosphate that you just broke off. That releases energy, somewhere in the range of 7.3 kilocalories. That energy can be used to do lots of things. It can be used for active transport. It could be used for cell division. It could be used for things like muscle contraction. Lots and lots of things. But you need to break that bond or hydrolyze the ATP. Well, where does the ATP come from? This here is the formula for aerobic cellular respiration. Aerobic having oxygen as your key ingredient. So you take in a glucose molecule, C6H12O6, 6 oxygens that you're going to get with the help of many enzymes. There's 6 CO2s, 6 H2Os, and somewhere around 36 to 38 ATPs. Well, what's going to happen here is these molecules, either the glucose or the oxygen, are going to be oxidized or reduced. So let's get rid of this for now. And let's start with the glucose. The glucose is going to lose its hydrogens, and it's going to become CO2. And what is going to happen there is you're going to lose electrons, aka you are oxidized. The O2 is going to gain those electrons, and that is said to be reduced. And we'll see where they gain them in a little bit. And I know it sounds weird to gain something and say reduce, but you're gaining a negative, so that's the reduced. So if we look at this equation, you have the molecule being oxidized as glucose and the molecule being reduced as oxygen. Now, how do you know that? Well, through radioactive isotopes, they did this in photosynthesis and in respiration. Let's say that they've radioisotoped that oxygen. Well, they found out later that that oxygen was this oxygen, not the oxygen released as from CO2. We'll see where that comes from in a little bit. So, which molecule in the products contains the original O2 that you breathed in? Well, it's the oxygen. And again, I'll show you how that works in a little bit. Actually, I'm going to draw the equation right now just so when we see it, it's not too crammed up. What's going to happen? They're going to use this equation of a half of O2, because remember the O2 comes in from breathing, plus 2 of these H pluses, and again, we'll see where they come from in a little bit, is going to yield for you water, which you will breathe out or pass from your cells into your blood. Well, the party all starts with glycolysis. Now, glycolysis is glyc, which would be kind of short for glucose. Lysis means to break. And it is the process of respiration that happens in the cytoplasm. Now, this is important because it is going to be known as an anaerobic process because it does not require oxygen. Well, we breathe oxygen and we do glycolysis. And anaerobic molecules can also do glycolysis. So basically what that means is we are breaking down the glucose in the cytoplasm by a process known as glycolysis in an anaerobic fashion, meaning we do not need oxygen to do so. Other organisms live entirely in an area that is considered anaerobic, and glycolysis is the only game they have in town. We will talk about those later. That's going to be alcohol fermentation and lactic acid fermentation. But because all cells can do this, it is basically stated that glycolysis is the universal energy releasing process, which suggests a common ancestor, which means that this has been going on in all living things since basically the beginning of living things. So this has been going on for billions of years. And the key here is this happened when there was no oxygen available in the atmosphere. And this happened before these organisms organisms did not have organelles. So therefore, things like mitochondria, they didn't exist yet in cells. Glycolysis is going to begin with what is known as the direct phosphorylation of glucose by substrate level phosphorylation. That is key. That means with the help of enzymes, you will be able to directly phosphorylate the ADP into ATP. Well, this is not going to happen unless you have kind of like a kickstart. I know this sounds silly, but glycolysis's job is to make ATP. Well, why the heck would you have to put some in? That to me doesn't make any sense. Well, like everything else, if you didn't have a little bit of energy to start the process, every time glucose entered your body, it would immediately be broken up, and that would be bad. So we have this glucose that enters your body through food or through breaking down of glycogen and moves through your blood and moves into your cells. Once it gets into your cells, then your cells will say, all right, let's start breaking this down. So they're going to do what is known as an investment of 2 ATPs. That is going to start the process of glycolysis, which again, we'll see in a second. And the payoff is going to actually be 4 ATPs. So you put in 2, you get out 4. That means you have a total net gain of 2 ATPs. And if you remember what I said earlier, anaerobic respiration is going to yield 2 ATPs. So if you are an anaerobic bacteria, for every glucose that you can, come in contact with, you will end up with 2 ATPs. You will also generate 2 NADHs. Now we're going to find in anaerobic respiration, these guys have a function. And in aerobic respiration, they have a function. But for now, let's just say glycolysis, 2 NADH, 2 ATPs. Now what we're going to find, as I draw the picture, to get this NADH, you need to start with something. It's called NAD+, which is going to be known as an electron acceptor. And we're going to see this NAD+ is going to show up a lot. It's going to show up in glycolysis, which we're going to talk about in a second, and then it's going to show up in Krebs cycle, which will happen in the next step if you are performing aerobic respiration. So, anaerobic respiration is going to happen in things like yeast, is going to happen in things like bacteria, and interestingly, muscle cells. Now you say to yourself, wait a second, muscle cells are part of our body, so, why are they doing anaerobic respiration? Well, we'll find out that if your muscle cells get kind of worked too hard, the available oxygen level goes down, and they have to figure out a way to generate some ATP, so we'll talk about that. Once again, anaerobic respiration is going to yield you 2 ATPs, and that is way, way, way less than what is known as aerobic respiration. And again, we're going to spend more time on that in a second. Well, the end result is start with a glucose, and you're going to ultimately end up with 2 pyruvate molecules. Now, when we move over to the picture on the next side there, you will see exactly what I'm talking about. So let's look. You start off with glucose, which has 6 carbons. There's your initial investment of the 2 ATPs, which is going to go into breaking up the glucose initially. And those 2 phosphates that you're going to steal off of there are going to end up right there. There's one, and there's the second one. That's going to be an intermediate molecule called glyceraldehyde 3-phosphate, or G3P. And this is, again, starting the process. With many enzymes and many intermediates, you will end up with 2 pyruvate molecules, 1 and 2. And here's where, things get a little interesting. There is that universal electron acceptor that happens here, this NAD, which is going to go into NADH. And here now, you're going to have two ADPs, and you're going to form two ATPs, and you're going to do the same thing over here. So there's your gain of 4 ATPs. 2 plus 2 is 4, but you have to subtract out the 2 that you started with, so there's your net gain of 2. This ATP that is formed through substrate-level phosphorylation. And all that simply means is with the help of enzymes, you can add a phosphate to a molecule. In this case, ADP is going to turn into ATP. Substrate level, enzymes needed, and that is 100% of the ATP produced from glycolysis is through substrate level phosphorylation. This also, can happen in anaerobic organisms, and you notice that because anaerobic means no oxygen, and throughout this whole process, no oxygen is needed. We will see how important it is coming up later, but right now, none is needed. So everybody can do it. That's evolution. And again, it happened in organisms before there was oxygen available and before any organisms had special organelles like mitochondria. But let's pretend we're talking about one of our cells now, and it doesn't matter which one. Stomach cell, it does not matter. In our cells, we have organelles, and these organelles are called mitochondria. Now, the mitochondria is pretty neat. You probably learned back in the old days of living environment or biology that's where your energy comes from. And, well, it does. And it's going to come in the form of ATP. So we are now going to start our next phase, which is known as the Krebs cycle. Now, Krebs cycle is important because what's going to happen is these pyruvates are going to enter the mitochondria, enter to the matrix of the mitochondria, and join up with the Krebs cycle. Well, like everything else, it's not that simple. Pyruvate has 3 carbons, as you can see from the picture. So there's 1, there's 2, there's 3. That's your pyruvate. And in order to get into the Krebs cycle, and the Krebs cycle is going to kind of be over here somewhere, you're going to have a 4-carbon molecule. It's going to be called oxaloacetate, or OAA. And you ultimately want to start forming this 6-carbon molecule called citrate. That's why the citric acid cycle gets its name. Well, we got a problem here because if the pyruvate tries to enter the Krebs cycle and you have a 4-carbon OAA and you try to join a 3-carbon pyruvate, that's 7. Well, citrate only has 6. So what do we got to do? Well, this is called the grooming phase, and what's going to happen is this pyruvate is going to do a couple of things. One, it's going to lose a CO2. Now, by losing a CO2, now we are down to 2 carbons, and we're going to gain this thing called a coenzyme A molecule, or CoA. So now you have a 2-carbon molecule called acetyl-CoA. So there it is, 2 carbons right there. And everything's great, but there's one more step. There's going to be you're going to take an electron, you're going to use NAD+, and you're going to convert that into NADH. Now, I've now said NADH several times. This NADH is going to become important because it can, if an organism has mitochondria, it's going to move into what is known as the electron transport chain and ultimately help us generate some ATPs. But right now, let's just be content with the fact that we've created a couple of NADHs. So this acetyl-CoA is a 2-carbon molecule, and it's got that little CoA molecule on top. Now, what's going to happen is when that joins the Krebs cycle, the CoA is going to get kicked out, and it will be able to eventually rejoin another pyruvate later, and that's just a beautiful cycle that happens just outside of Krebs cycle. So now you have your 2-carbon acetyl-CoA minus CoA plus your OAA equals citrate. And that is going to continue in a cycle, and we'll see more in the Krebs cycle on the next page. But this whole thing is known as the grooming phase. And you can kind of think about it as when you take the dog to get groomed, it gets cut up. And here, the grooming phase is you are cutting off a CO2, and that's the grooming phase. Pyruvate cannot enter the Krebs cycle by itself. It needs to be converted into acetyl-CoA first. Next is a very complicated diagram, and that is the Krebs cycle. Again, you will absolutely not need this in all the detail, but let's look where we are so far. There's my pyruvate right there, and this is the grooming phase, which we talked about already, and the grooming phase is going to minus 1 CO2, add 1 CoA. So there's the CoA right there, joining the party. There's my NAD turning into NADH, and there's my loss of my carbon dioxide. There's my 2-carbon acetyl-CoA, which is going to join my 4-carbon OAA, and there's my 6-carbon citrate. Now here's where things get interesting. There are a lot of intermediates. You will not need to know any of them, but here's what you will need to know. Along the way, you're going to see that these molecules, all of these intermediates, are going to be oxidized along the way, and they're going to get oxidized to form NADH right there, NADH right there. You're going to form something called, there's the other NADH way over here, NADH. And along the way, you're going to also form an FADH2. I don't know where that is on this diagram, but let's draw it over here. So you're going to have an FAD that's going to go into what is known as an FADH2. Now these things are important because they are carrying electrons, and they're going to take them to this place called the electron transport chain. You're also going to have this little molecule of GTP, and as far as you're concerned, GTP is equal to ATP, so it's close enough. And that's going to happen by a little substrate level phosphorylation there. So you're oxidizing your intermediates as you go. You're generating these things called NADH, these things called FADH2. And as this cycles around and around, these NADHs and FADH2s are free to leave. Well, where are they going to go? It's a great question. They're going to head off to what is known as the electron transport chain. But let's do a little review here. So, pyruvate is going to get converted into acetyl-CoA. We talked about that. It's a 2-carbon molecule. It is now able to enter the Krebs cycle, and this is all going to happen in the matrix of the mitochondria. So the grooming phase, which we talked about, you're going to add a CoA, you're going to lose 1 carbon dioxide, and you're going to turn an NAD plus into an NADH. Now, I'm going to tell you in a second that you're going to multiply all of these numbers by 2, because remember, 1 glucose makes 2 pyruvates. And in this chart so far, we started with only 1 pyruvate. So we got to double it because we have 2 of them. Once you get into the Krebs cycle, you are going to produce 3 NADHs, 1 FADH, 1 GTP, otherwise known as ATP, and you're going to kick out 2 carbon dioxides. So here's where things can get tricky. If they talk about the process from pyruvate all the way to the end, you have a total of 4 NADHs, 3 carbon dioxides, 1 FADH2, and 1 GTP, which of course you're going to multiply by 2. If the question is specific and asks you only about the citric acid or Krebs cycle, you must remember that the grooming phase happened before you got to the cycle. So it is important, read the question carefully. All of these numbers you see are for 1 pyruvate, but remember, there are 2 pyruvates. So when you do your final total, that's why we multiply everything by 2. Now for the fun part. All of this work that we have put in is going to go into this diagram right here, which is known as the electron transport chain. I'm going to describe the picture first, then we're going to go through all of the words. Very important to know where the heck we are. So if this is the mitochondria, and this is your standard issue mitochondria, That is the outer mitochondrial membrane. That's the outer mitochondrial membrane, which means this area over here would be considered the cytoplasm. And this is where, of course, glycolysis would take place. Then you have this thing, which is the cristae, or the inner mitochondrial membrane. This area here is your matrix. And then this area that's kind of in between the inner mitochondrial membrane and the outer mitochondrial membrane is known as the intermembrane space. So let's see where we are. This up here is your outer mitochondrial membrane. This is your inner mitochondrial membrane. The area between them is known as the intermembrane space. That's where my dot is, right over here. And the matrix would represent this area right there. So what the heck is going on? Well, in the inner mitochondrial membrane, there is going to be a series of, we're going to call them proteins, we're going to call them protein pumps. And what they are, otherwise known as the cytochrome network, is they're embedded in the inner mitochondrial membrane, and they have one well, they have a couple purposes, but one of the purposes they're going to do is they're going to take that NADH, if you see that over here, that NADH that was created in either glycolysis or the Krebs cycle, once again in glycolysis and aerobic respiration, it's then going to join up with this cytochrome network, or this series of membrane-bound proteins. And what it's going to do is it's going to come in and it's going to do kind of like two things. One, it's going to drop off its electron. That's right here. And it's going to have an H+ that's going to kind of be chilling out here in the matrix. Well, this electron has a lot of energy, and that energy is going to pass through this first pump, and as it passes through, the hydrogens that are in the matrix are going to pump through into this area known as the intermembrane space. So you're going to start pumping hydrogens. Well, this electron, let's follow it through, is then going to go through a second pump right there, where this hydrogen is going to be pumped into the intermembrane space. It's then going to go through a third protein, where it's finally going to come to its friend oxygen, which is known as an electron acceptor. All the while, as it passed through 3 pumps, it moved 3 hydrogens from the matrix into the intermembrane space. This is active transport. It's active transport because I added energy and I went from a low concentration of hydrogens into an area of higher concentration. So in essence, this intermembrane space is going to start to fill up with these hydrogen ions, otherwise known as protons. And with this huge influx of protons or hydrogen ions, you're going to build up some pressure. And inside of that area, all of this pressure is going to be known as proton motive force. So the proton motive force is in essence all of the pressure from these protons. Well, they have to be released somehow, otherwise, the mitochondria would be in trouble. So they're going to be released through this kind of other protein called ATP synthase. Now, this is very, very, very important, because what's going to happen here is now the H's are going to start to come down back into the matrix. And what I mean by down is now they're going from a high concentration to a low concentration. So in essence, they're going with their concentration gradient or along their concentration gradient. And because they're doing that, now this is not a pump. This is actually, this is diffusion. So because you're coming through this ATP synthase, it's going to be called facilitated diffusion. And it's going to have an even more fun name called chemiosmosis. Did I spell that right? Chemiosmosis. Nope. Extra O. Oh man, this is embarrassing. CHEMIOSMOSIS. O-S-I-S. Always fun when you spell something wrong in a video. Chemiosmosis is the movement of these protons from high concentration to low concentration as they scream through this protein called ATP synthase. And when they do that, they are going to take the ADP plus a phosphate that's in the matrix, and you're going to generate some ATP. And you're not going to generate some ATP, you're going to generate a lot of ATP. So, which we're going to find out in a second, for every NADH that passes through 3 pumps, you're going to move 3 hydrogens. If you move 3 hydrogens, those 3 hydrogens are going to come back through ATP synthase, and they're going to generate 3 ATPs. Well, our friend FADH2 is going to do the same thing, but that's going to skip one of those proteins. So you see here it kind of jumps on the party a little bit late. And it's going to go through 2 of these pumps. And if it goes through 2 pumps, it's going to move 2 hydrogens, and therefore it will make 2 ATPs. So we'll see that in a second. But either way, they're going to crash into this oxygen. Now, oxygen is known as and you can see it right here, final electron acceptor. So because of that, because the oxygen had the ability to pull this electron through the chain, you were able to pump the hydrogens against their concentration gradient. They were then allowed to come back through the ATP synthase and generate this ATP. This is called oxidative phosphorylation because none of this would have happened without oxygen. Now, the oxygen does not get involved in actually phosphorylating. It's not needed to actually put the ADP and the phosphate together. But without the oxygen, the electron is not driven. And without the electron not being driven, you don't pump the hydrogens. You don't pump the hydrogens, you don't generate the ATP. So that's why this is called oxidative phosphorylation. Now, this whole process, known as the electron transport chain, does not need any heat to get started. It will release heat as a waste product, but no heat is needed to start this party off. This will happen as long as oxygen is present. So let's now kind of go through all the words that we just talked about. So the electron transport chain, let's kind of put this over here, get out of the way, is going to take the NADHs from either glycolysis in an aerobic organism like us or through the Krebs cycle, and the FADHs from the Krebs cycle, and it's going to move them into the inner mitochondrial membrane into what's known as the electron transport chain. The NADHs, we said, will generate 3 ATPs. The FADH2s will generate 2 ATPs. And the way I remember it is that 2 and that 2, kind of easy to remember. And this is going to happen in a series of proteins, which we said, called the cytochrome network. So you may see that. And those proteins are going to turn into pumps. And since they're pumps, that's going to be known as active transport, which is going to cause an unequal distribution of hydrogen ions or an unequal distribution of protons. So because of that, you're going to now have a concentration gradient going on, and the hydrogens can move from a high concentration to a low concentration through ATP synthase. And when that happens, your ATP will be generated. A lot of ATP will be generated. And you may see the words proton motive force. That's the force of the hydrogen ions or protons moving through the ATP synthase. Or you may see the word chemiosmosis. Those are words related to hydrogen moving through the ATP synthase, generating ATP. Once again, this is facilitated diffusion, not a pump, because it is moving from high to low through this protein, generating the ATPs. And the fact of this is there's a large pH difference between the inner mitochondrial membrane and the matrix because of all of these hydrogens. And once again, hydrogens and pH are pretty much related. So that is glycolysis, that is the Krebs cycle, that is the electron transport chain. So we covered a lot of aerobic respiration material. Now let's move our story to anaerobic. Now this is where things are going to get a little bit tricky, so let's kind of take our time here. Anaerobic respiration happens when oxygen is not available, or it's in short supply, or the organism does not have mitochondria. So something like bacteria. They don't have mitochondria. So let's start with lactic acid fermentation. And that's going to happen in a couple of places. That could happen in our muscle cells. And again, we'll just stick with me for a second. And that's going to happen when we are in oxygen shortage. So obviously you are aerobic, but if you exercise very, very, very strenuously, you may come in times of low oxygen and your muscles still need ATP, so they're going to kind of switch over to lactic acid fermentation. And in us, in humans, this lactic acid, when it builds up, it's going to hurt tomorrow. So if you've ever worked out kind of hard and then the next day you were sore, it's lactic acid. We may also see this in the bacteria that produce yogurt. So those are the two places that lactic acid fermentation will happen. Now this is very, very, very important. You start off with glucose, which is a 6-carbon molecule, which again, we must have said that a bunch of times. And you're going to go through this top process. So this top process here is glycolysis, and you know that. Whoops, you know glycolysis. Glucose to 2-pyruvates, and let's get rid of that. Let's kind of do a better box there. Glucose to 2-pyruvates is going to yield you 2 ATPs and 2 NADHs, and everything's great. Now, under normal circumstances, these pyruvates are going to go off to the matrix and life is good. That's what would happen under normal circumstances. But if a human exercises too much and the muscles kind of go into a slight little bit of anaerobic respiration, let me get rid of this box right here. And now the pyruvates that you created do not go to the matrix. They stay in the cytoplasm, and what they're going to do is convert into what's known as lactate. Well, to do that, this is the key. The NADHs that you produce that would have normally gone into the Krebs cycle are going to stay back in the cytoplasm. And what's going to happen is you're going to do a little reduction right there. And you're going to take the H and you're going to take the H off, and you're going to form two NAD plus molecules. Now, this is why this is super duper important. Now that the NAD plus is generated, can it act on another glucose to at least get you 2 ATPs, forming pyruvate again, forming more lactic acid. So the longer you exercise in times of low oxygen, the more lactic acid that builds up. So the more strenuous your activity, the more you will hurt tomorrow. Once you've rested and you're back to normal, this process shuts down. But unfortunately, the lactic acid will stay in your muscles for a couple of days and it will hurt. So that stinks. So the regeneration of NAD plus is mucho, mucho, mucho important, because if you don't, the process of glycolysis again will stop. So very important, regenerating that NAD plus and not letting that NADH go into the mitochondria. So in humans, which we just said, if you exercise very, very, very strenuously, you're going to go into lactic acid fermentation. And what's going to happen is your lactic acid is going to increase. Ouch. Your NAD plus is going to increase, which is good, so at least you can generate some ATPs. And any glucose you have is going to go down, so hopefully you ate. And if you didn't, your glycogen levels are also going to go down, because if you don't have any readily available glucose, your body's going to go and break down glycogen. So, in exercising strenuously, lactic acid builds up, ouch. NAD plus is available again, so at least you can generate some ATPs. Glucose goes down and glycogen levels go down. After a while, if you exercise too hard, you may go into muscle fatigue and fall down, and that would be bad. So again, if you're going to exercise, eat right, drink a lot of water, and don't overdo it. What if you are something cool like yeast? Now, yeast is not going to form lactic acid. Yeast is going to form something that we, as humans, enjoy, if we're old enough. And this is alcohol fermentation. It's going to start exactly the same way as lactic acid fermentation. You're going to start with glucose. You're going to generate a total of 2 ATPs and a total of 2 NADHs. Now, these guys probably do not have mitochondria, so you can forget about that whole matrix thing. So, in order to keep on breaking down glucose to get energy, we have to, again, swing around, but we're not going to make lactic acid this time. We're ultimately going to make alcohol in the form of ethanol. And we're going to kick out a couple of carbon dioxides each time we do this. So if you think about bread, when you cook it, there's yeast, and the yeast is living off of the carbs in the flour, and it's going to generate CO2. So if you ever let kind of dough sit, it will rise. That's the CO2 formed from the yeast. And when you cook bread, it's full of air pockets. Those air pockets are the CO2 pockets. And the reason why you don't, get intoxicated from bread is because as the bread cooks, the alcohol will burn off. So this is anaerobic respiration, alcohol fermentation, and lactic acid fermentation. So there are similarities and there are differences, and we know which organisms each occur in. And the last and final part of this story is what's known as thermoregulation. So we have been dealing a lot with respiration and cell respiration and so on. And we are going to be using oxygen, consuming oxygen, and we are going to be producing carbon dioxide. And that's what happens in cell respiration. So if we had really fancy tools, we could kind of put an organism in a box and we could measure how much CO2 it produces or how much O2 it uses, and that would be great. And the rule of thumb is the more O2 you are consuming, the more ATP you are making, or the more or greater your respiration rate is. And that should make sense. And since we're dealing now with animals, this is all going to be through what's known as oxidative phosphorylation with the electron transport chain, just like we talked about. So we talked about a couple of things in class. We talked about endotherms, which would be your mammals and your birds. You may know them as warm-blooded, but we're not going to say that. We're going to call them endotherms. And ectotherms, which you may know as cold-blooded, which again, we're not saying that either. That's going to be insects, amphibians, and reptiles, and some fish. And these guys, ectotherms, their body temperature is affected by the external temperature, where endotherms, their internal temperature is reasonably constant. So the major difference here is how they regulate their internal temperature. That's basically our story. So endotherms, let's start with us, are going to keep a reasonably constant body temperature. And again, that means under normal circumstances, getting thrown into the freezing cold ocean in your Speedo, that doesn't count. Just normal environmental areas where you would live comfortably. So if the external temperature drops, let's say you're outside in a t-shirt or whatever, and it's, now, November-ish, in order to maintain your warm internal body temperature, you got to start to do something. You're going to start to contract some muscles, and you're going to try to generate some ATP. And we call that shivering. So as our outside temperature drops, we try to maintain our internal temperature by shivering. So we're going to have ATP production, we're going to have oxygen consumption, we're going to have a high respiration rate, and with all that respiration, we're going to have a waste product of heat, which hopefully will warm us up. Well, the exact opposite will happen if we are in a very warm and comfortable environment. Let's say that we are outside in a nice 85-degree day with light winds and the sun's out. In this case, we don't need to generate excess body heat. Our respiration rate, our O2 consumption, and our ATP production are going to go down. So it's kind of neat how it's opposite of what you might think. So if the organism gets way, 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 way too hot, then things kind of get all weird and we begin to sweat. But let's just keep it simple that as the exterior temperature changes, as it gets colder, our ATP production goes up. As it gets warmer, our ATP production goes down. An ectotherm is, they got a little bit of a bigger problem there. Their body temperature fluctuates with the external temperature. So if it's really cold out and you're a lizard or you're a reptile, you're going to slow down to the point of you might even die, because your metabolic processes are going to slow down so much. And if your metabolic processes slow down, you're going to consume less oxygen. If you consume less oxygen, you're going to produce less ATP, and you're going to have a lower respiration rate. So their body temperature is directly related to the outside temperature, and their respiration rate is related to the outside temperature. So if we check out how the heck we could even measure all of this, we use something called a respirometer. Now, what they did was they were measuring oxygen consumption, because how they did it in the old days. They didn't have fancy probes and whatnot. And what they did was they took an organism. In this case, it was a cricket or a grasshopper or something, but whatever. You put anything in this tube. And what you do, and what we did in this lab we put a drop of water right there. Now, what's going to happen is, since that drop of water is there, it blocks the intake. So this cricket here is going to take in oxygen like that, and you could put anything in there. You could put a mouse in there, a cricket, whatever. And it's going to release CO2. Now, if it takes in oxygen and releases CO2, the volume of air inside of that chamber is not going to change. So scientists were like, okay, what do we need here? Well, we need a chemical. So they came up with this potassium hydroxide, and they soaked it in a little bit of a cotton ball. And what happened was, every time the organism breathed in oxygen, that was great. They breathed out CO2, that was great. This CO2 then went to the potassium hydroxide, and it turned into a solid precipitate. Now, because of that, as this organism breathes, you're going to end up with less gas in the chamber. That's going to cause a vacuum. So what happened was, this little droplet of water began to move down this kind of like little ruler, or this little graduated cylinder. And what you were able to do was measure the change in volume. And in this case, the change in volume was oxygen. So you were able to do oxygen consumption. And the more crickets you put in there, the more oxygen they consume. The bigger the animal, like a mouse or a chipmunk or something, the more oxygen was consumed. And this was a really kind of nice way to actually see it happen. And then they started to change the temperatures, and before you know it, here's what they found out. Well, newer technologies, of course, you can use probes, but we're going old school here. So I use green and I use red. So green is going to be your endotherm, and red is going to be your ectotherm. So the first thing they did was they said, all right, let's see. We had some graphs, which again are all modes of thermoregulation, and they said, all right, if we change the external temperature, and again, we didn't go crazy. Let's say we made this, let's call it 60 degrees Fahrenheit all the way up to, let's call it 90 degrees Fahrenheit. And again, that's not extreme, it's reasonable. What happened there, your endotherms, again, your birds and your mammals, their body temperature remained fairly constant. But your ectotherm, what they found was, as the external temperature increased, so did the body temperature of the ectotherm, things like reptiles, amphibians, and things like that. So in the event that you have a snake cage at home, or you have lizards or any of that, you're going to have a heat lamp in their cage, because they're going to be way more active if they're warm. And again, if your room gets too cold, you may find it comfortable at 60-something degrees. They're freezing. So it's very important to keep that in mind. Now, again, this is not going to the extreme of, 150 degrees Fahrenheit. This is just normal, standard conditions. And then this is what they found with respiration, which was kind of neat. Using the respirometer, they said, how does O2 consumption, aka otherwise known as respiration rate, otherwise known as ATP production, what happened? Well, they said that in the cooler temperatures, in the 60 degree Fahrenheit range to the 90 degree Fahrenheit range, just keeping consistent, that your ectotherms, as you increase the temperature, they increased oxygen consumption. They saw more or higher respiration rates. They consumed more oxygen. Your endotherms, let's say again you put a mouse or something like that in the chamber, they found out that as it got warmer, they consumed less oxygen. They had lower respiration rates. They produced less ATP. And all of this was, fairly obvious once you set up a graph and you put in the respirometer and you measured all these variables. And then it became crystal clear once fancy probes came out and they could measure oxygen and carbon dioxide at the same time, and you didn't need the KOH, and it was pretty cool. So that is all respiration, both anaerobic and aerobic, the parts, glycolysis, Krebs cycle, electron transport chain, and thermoregulation. So I hope you thought that was helpful. Good luck.